And I know that you folks in the Center for Population Biology think about this tree a lot. Finally, so how do I advance? Okay. So we're actually here. So finally, this population is made out of individuals. And within individuals, there's a third type of tree, the cell tree, the branching pattern of cell division. So the developmental biologists in my department tend to pursue this tree. So these are three very disparate fields of biology. But what if I told you that life is branching all the way down? There is a nested recursive pattern of trees stretching from the macro scale of the species all the way down to the micro scale, the level of the cells. So these trees are expression of the same phenomena at different biological levels of organization. So today I'm going to be talking about branching patterns at one of the smallest scales, the level of the cell. So there are three trees that broadly describe cell type diversity. So for centuries, people have categorized cells by phenotypic similarity, which forms a nested hierarchical pattern. This is the cell taxonomy. So just like the traditional species taxonomy of the 18th century, this is not an evolutionary or developmental tree, it's a description of phenotype. A second category of trees is the cell developmental lineage, the branching pattern of cell division. So developmental biologists have pursued this tree since the 1800s. But there's a third tree, and this one is much less studied, and that is the cell phylogeny, the evolutionary history of cell types. So cell phylogeny is a little bit mind-bending, so let me walk you through it. So what is a cell phylogeny? A cell phylogeny is just like a species phylogeny, except the taxa are cells instead of species. It maps the evolutionary history between cell types, and it shows the pattern by which an extant cell type was inherited from an ancestral cell type that existed in an ancestral organism. So for example, when we say that muscle cells in a mouse and human are homologous, what we really mean is that on the cell phylogeny, they both descended from the same ancestral muscle cell type that existed in a common ancestor to mouse and humans. But we can also identify more distant homologies. For instance, the rod and cone photoreceptors in the eye, they're really divergent morphologically, but we know they probably descend from the same photoreceptor cell type. And this probably happened about 500 million years ago, given that jawless vertebrates also have rods and cones. So I want to make a really clear distinction between the cell developmental lineage and the cell phylogeny, because these two trees are really easy to mix up. So in a developmental lineage, the tips are differentiated adult cells. The interior nodes are precursor cells that have since divided during embryogenesis. So branching depicts cell division that happened during the lifetime of an individual, and the root is the zygote. In contrast, in a cell phylogeny, we have extant cell types at the tips, and the nodes represent a cell type that existed in an ancestral organism. So the scale of this tree spans millions of years, and the root is a single ancestral cell type. But what's the point of having cell phylogeny? So I think there are many good reasons, but one reason is that cell phylogeny gives us a concrete, direct phylogenetic framework to pursue some of the biggest questions in biology. So a really obvious one is the origin of multicellularity. The cell phylogeny is a depiction of evolution from the first ancestral cell type to technically every cell alive today. So theoretically, a cell phylogeny can map the origin of animals. Another classic question is homology between cell types. So let me introduce you to a story. So chronoflagellates, the closest unicellular relative to animals, and sponge chronocytes, the sponge feeding cell type, have long been assumed to be homologous. And that's because they show this really striking morphological character, which is that collar or microvilli surrounding a flagellum. So this has been used as a basis to argue that sponges are sister to the rest of metazoa. But we can see that recently, morphological, olfactory, and transcriptional data suggest that maybe these two cell types are convergent. So we can answer this question with the cell phylogeny. Do these cell types form a clade? So we can go further and examine the evolution of the collar itself through an ancestral character state reconstruction on the cell phylogeny. Finally, the last example I'll leave you with is the question of complexity. So why do some organisms, like Trichoplaxid heron, have only about 30 cell types, but other organisms like us, we have over 500 cell types? So people have taken many approaches to this question, genetic, developmental, but from a phylogeneticist point of view, this is a question about heterogeneity in the rate of cell type evolution. So when we think of complexity, we are so used to mapping the number of cell types to the tips of the animal tree. But in my opinion, what this creates is a scala naturae. So let's take the perspective of a cell. So maybe the emergence of a new cell type is equivalent to speciation, the emergence of a new species. And if so, we can use the same phylogenetic comparative tools we use to model speciation to model cell type diversification. So maybe more complex animals experience an acceleration in the rate of cell type diversification. And this is a completely orthogonal approach to the traditional way that we currently study complexity. So there's been a long history of study of the cell taxonomy, and there's been a long history of study of the cell developmental lineage, but much of this work has been done on the cell phylogeny. And why is that? Well, for a long time, we didn't have access to the amazing amount of cell type diversity that we have today. So traditionally, cells were studied by tracing and labeling them one by one. But now we're in the single cell era. So single cell sequencing works like this. You take an organism and you grind it up into a suspension of single cells. Each cell is captured in a single oil droplet with a special bead. The cell is broken open and each RNA molecule attaches to the bead to get a special barcode. This barcode has unique identifiers that lets you determine which molecule and which cell that transcript comes from. And finally, we sequence each cell using next generation sequencing, and we count how many transcripts were present for each gene in each cell. So these RNA counts are the cell transcriptome. So ultimately, this produces a single cell counts matrix. So in this matrix, each row is a gene and each column is a cell. And the values are the number of transcripts expressed per gene in each cell. So for example, in cell 1, gene A expressed 1 transcript. These gene expression matrices tend to range from about 5 to 10,000 cells and about 20,000 genes. And they're often very sparse. So that's why you see a lot of zeros, ones, and twos. So it turns out that a gene expression matrix can actually make a pretty good phylogenetic character matrix. All you have to do is flip it on its side. So in this case, now the rows are cells, which you can call taxa, and the columns are genes, which you can call phylogenetic characters. So this produces a matrix that is suitable for phylogenetic analysis, but there's something different about these matrices compared to most modern phylogenetic matrices. The thing is, most modern matrices are built using sequence data, especially DNA sequence data. So sequence data is discrete, but within any single organisms, cells don't differ by their genome, they differ by their transcriptome, and a transcriptome is continuous data. Continuous characters come with their own issues. So in the following section, I'll demonstrate how I approach these challenges to produce the first cross-species cell phylogeny. So today I present to you a phylogenetic analysis of cell types. So for part 1, I'll present a method that I published in this paper, Cell Type Evolution Reconstruction Across Species, from cell phylogeny of single cell RNA sequencing data. It's a method to build cell phylogenies from single cell count data using an explicit model of evolution. So let me walk you through this. I'm going to walk you through the 15 steps it takes to make a cell phylogeny. Hey, I'm just kidding. But I'm going to describe the major conceptual breakthrough needed to make this workflow work. So most phylogenies are built using discrete data, especially DNA sequence data. But DNA sequence data really only varies in 4 ways: A, T, C's, and G's. So the number of parameters you need to calculate increases linearly with each discrete character. But again, within any single organisms, cells don't differ by the genes they possess, but by the genes they express. And gene expression is a continuous character. So most models of continuous character evolution, I'm not sure why it's cut off. But most models of continuous character evolution were actually invented back in the 1970s, back when the data were things like gene allele frequencies and morphology. But these models do not handle covariance well. And the issue is gene expression is rife with covariance. So this has a number of implications for the number of parameters that we have to calculate. So phylogenetic models of continuous character evolution have to calculate something called a variance covariance matrix. So here's a diagram of one here. The variances in blue are the diagonal, but in red on the off diagonal are all the covariances you have to calculate. And there are many more covariances than variances. In fact, for each additional continuous character k, you have to calculate k squared minus k over 2 covariances. In other words, the number of parameters with each continuous character increases quadratically. So that really gets out of hand. So for 20 characters, you have to calculate 230 parameters, but for 20,000 characters, the size of a gene expression matrix, you have to calculate over 200 million parameters, and that's not computationally feasible. So this is called the NP problem. We were told that it's theoretically impossible to infer phylogeny from highly multidimensional continuous data. That's because n, the number of samples, isn't high enough to calculate p, the number of parameters. There simply isn't enough information in a single cell count matrix to infer a robust cell phylogeny. Or what if I told you that we can zero off all the covariances and keep just the variances? So not only we can do that, but we can do it with a method that I think everyone in the room is probably familiar with: principal component analysis. So I won't get into the mechanics of PCA, but PCA parses out patterns of variance and rotates the data onto new axes of variance called principal components. So principal components are independent and orthogonal to each other. In other words, they don't have covariance. This means we can drastically lower the number of parameters that we have to calculate if we use principal components as phylogenetic characters. So I got really excited. I thought, okay, I've finally found an approach to the NP problem, and we can totally infer cell phylogeny. So we did. And this is what I got: a perfect comb. Honestly, this tree is so comb-like, it doesn't even look like a tree. So each line is a tip branch that stretches from the root of the tree all the way to the tip. So if there was any phylogenetic signal, it would be somewhere here. So the rotation was necessary to make the characters independent, but it looks like it pushed all the variance to the tip branches, leading to a star phylogeny. So there are many things that can affect gene expression in a cell. The first is the cell type identity. That's the cell phylogenetic signal that we want to isolate. But there's also cell-specific variants, things like sequencing noise, physiological reactions, cell cycle state. So what if the cell phylogenetic signal is there? It's just being obscured by cell-specific variants. So here's a plot of the amount of variance described per principal component. And the first thing we notice is that the data is very low rank. You can see that 27% of the variance is described by just the first 20 PCs. There's an additional property of PCA. PCA sorts variance hierarchically in descending order. So the earlier principal components describe broad, low-frequency signal, whereas the later principal components describe fine, high-frequency signal. So I hypothesized that the cell phylogenetic signal is a broad, low-frequency signal, and in contrast, maybe cell-specific variance is a high-frequency signal. So that means if we truncate the number of PCs, we can remove all the PCs that represent cell-specific variants and keep just the PCs that represent low-frequency phylogenetic signal. So this arrow plot suggests that it's appropriate to just use the first 20 PCs. But is the first 20 PCs where the phylogenetic signal resides? So to study this, we performed a sweep through the principal components. So we wanted to understand what kind of variance is contained within each principal component. So we created about 98 matrices where we increased the number of principal components from 3 to 100 principal components. And we then inferred the trees and plotted the results. So this, we studied the data in a couple ways, but this is the most important plot of the paper, so I'm going to zoom up to it. So this graph shows the sum of the interior and tip edge lengths of the trees that we inferred. So in a phylogeny, phylogenetic signal is present in the shared interior nodes. In contrast, tip or cell-specific signal is present at the tip edge lengths. So we see that at approximately 20 PCs, tip edge length increases, but interior edge length stays about the same. So this means that past PC20, much of the variance is actually tip or cell-specific, which suggests that the phylogenetic signal may instead reside in the first 20 PCs. So when we use 100 PCs, we get a comb. When we use 500 PCs, we get that perfect comb. When we use 100 PCs, we start to see some phylogenetic signal, but when we only use 20 PCs, we finally get the emergence of true cell-type clades, and that is a beautiful cell phylogeny. One thing I'm not going to get into is how I calculated support values. So we couldn't use Bootstrap, because Bootstrap assumes that all the characters are weighted equally. But principal components are weighted in order of descending variance, so Bootstrap wasn't appropriate. So I invented two new measures of repeatability, jumbo scores, which measure technical repeatability, and single-cell jackknife scores, which measure biological repeatability. But for brevity, I'm not going to get into them, so if you're interested in them, I'd be happy to talk to you about them after the talk. So here you go, here's our cell phylogeny. So I'll get into more detail about the pilot data set we used, but just know that it consisted of a set of eye cells that were dissected from 5 mammalian model species. So I was overjoyed to see that cross-species cell-type clades emerged. Not only that, but they had high technical and biological repeatability, both between and within cell types. But what was interesting is that we've attained a phylogenetic definition of cell type, as has been theorized for over a decade. A cell phylogeny changes how we think about and define cell types in a similar way to how a species phylogeny changed how we think about and define species. So to recap, we found solutions for 3 big technical challenges. I approached the MP problem by using principal components as phylogenetic characters. I isolated the cell's phylogenetic signal by keeping early PCs and discarding later PCs. And finally, I developed 2 new measures of repeatability because Bootstrap wasn't suitable. Okay, so that was all super technical. But what about the biology? I am a biologist, and I do believe that a phylogeny is pretty boring if it can't tell us anything about biology. So I'm going to walk you through a couple of rich biological stories I found in the phylogeny. Let me introduce you to my pilot data set. It consists of eye cells from 5 mammalian species, a human, 2 monkeys, a mouse, and a pig. The cells came from the aqueous humor, so eye fluid, an anterior segment of the eye. And this eye has a lot of channels to move eye fluid around. So the first thing we did was examine what our principal components represent. We did a gene ontology analysis of the most highly loaded genes and found that they seem to describe cell type specific signals. So this is a table of the ranked most positively loaded genes. And I've highlighted the genes in blue that appear to contribute to a cell type signal. For example, PC2 produces vasculogenesis coterms, and we find that PCAM1, the canonical vessel endothelium marker, is the 6th most highly loaded gene. So the PCs have a biological logic to them. They describe broad patterns of gene expression that define the function and identity of these conserved cell type clades. So let's take a closer look at the tree. Let's start with the Schwann cells. So Schwann cells are a nervous system type of cell that wraps around neurons and produces the myelin sheath. The topology of the cell tree lets us infer specific evolutionary events that happen during cell type evolution. So like in a gene tree, some interior nodes represent divergences between cell types. Other interior nodes represent divergences between species. So the Schwann cell clade is a really clear example of this. The black node is a within cell type divergence, an ancestral Schwann cell type program split into the myelinating and non-myelinating subclades, but the white nodes are divergences that happen with speciation events. So the sister relationship between myelinating and non-myelinating Schwann cells suggests that they are sister cell types, which is a similar concept to sister groups on a species phylogeny. We found another interesting pattern in endothelial cell types. So there were 2 types of endothelia on our tree. There's the corneal endothelium, which is epithelial cells that line the base of the cornelium in your eye. And then there's vessel endothelia, which line blood vessels. These 2 types of cells are classified as endothelia for a reason. They look very similar. So why didn't they group on the cell tree? Well, it turns out that vascular endothelia actually comes from mesoderm, whereas corneal endothelia comes from ectoderm-derived neuropress. And this suggests that maybe the two cell types are convergent. Let's take a closer look at the vasoendothelial clade. So in this clade, there are three subtypes. There's the vascular endothelium, the collective channels, and the Schlemm's canal. So the Schlemm's canal and collective channels drain eye fluid from the eye. They have a very intimate morphological and anatomical relationship. The collective channels are actually continuous with the Schlemm's canal. But our cell phylogeny says that the collective channels are actually more closely related to vascular endothelia, not the Schlemm's canal. And why is that? Well, there are two vascular systems in vertebrates. There's the lymphatic and blood vascular system. So during embryogenesis, the blood vessel system can transition to a lymphatic fate through the expression of lymphatic marker genes. And in fact, recent research indicates that the Schlemm's canal actually expresses these lymphatic marker genes, whereas the collective channels only express blood vascular marker genes. So it is possible that the topology of the cell tree reflects this venous versus the lymphatic split. Finally, the last story is the sister relationship between the ciliary parasites and the ciliary muscle and the parasites. So these two cell types have very different anatomical and functional roles. The ciliary muscles controls lens movement, whereas the parasites wrap around blood vessels. But the phylogeny suggests a close evolutionary relationship. So let's look at the biology of these cells. Parasites really aren't similar to ciliary muscle, but they are pretty similar to smooth muscle. Both smooth muscle and parasites are contractile cells, and they both express the marker gene ACTA2. Similarly, ciliary muscles is not like parasites, but it also kind of is like smooth muscle again. It also expresses ACTA2. And the canonical smooth muscle regulators, nitric oxide and endothelin-1, actually control both ciliary muscles and parasites. So these molecular similarities suggest a close evolutionary relationship as supported by our cell phylogeny. Okay, so that brings us to the end of part one, an introduction to the cell phylogeny method. What did we learn? We found that yes, we can infer cell phylogeny from the eye cells of 5 mammalian model species, and this tree produced novel insight into the origin and evolution of eye cells. So for part two, I take my method out into the real world. This is my pilot data set. They're all mammalian, and they all come from a single organ. So can I use this method with more distantly related cells? So here I present a study on the evolution of cell types that arose about 450 million years ago. Okay, so I've got this exciting new method, but the first question everyone asks is, does it work on anything that isn't your pilot data set? What is the taxonomic scope of my method? So I decided to look at 3 phylogenetically nested clades. The first is Planulozoa. That is the clade consisting of Cnidaria and Bilateria. So Cnidaria are the sea anemones, jellyfish, and corals, and I think everyone here is familiar with Bilateria. So I had 6 Cnidarians, 3 amphizoans, and 3 hydrozoans, and then for the Bilaterians, I had a human, a fly, and the flatworm Schmidtia. The second clade I looked at was Cnidaria, and I looked at 3 amphizoans, which is Nematocilla, Stylophora, and Xenia, and 3 hydrozoans, which is Hydra, Hydractinia, and Clidia. Finally, the third was the most restricted clade, which was just Hydrozoa, a clade within Cnidaria. So as just mentioned, this includes Hydra, Hydractinia, and Clidia. An important biological question is the evolution of stem cells. Stem cells are a type of cell type that exhibits potency. And potency is the ability of a cell to differentiate into other cells. So there are many types of stem cells in the clades I just mentioned. Humans have embryonic stem cells. Schmidtia has like these adult pluripotent stem cells. But the remarkable stem cell system of hydrozoan cnidarians may give insight into the origin and evolution of animal stem cells. So I tried the analysis, and the first thing I found was that Planulozoa is too distantly related. You know, Planulozoa is an extremely deep clade. It's about 650 million years old. So what I didn't get into earlier is there's actually a step you have to perform before the inference of the cell phylogeny, and that is cross-species integration, because this removes the species signal and aligns cells across species, so that the variance that comes with being different species doesn't obscure the variance that comes with being different cell types. So here, cross-species integration fails because there simply aren't enough genes universally shared across all these species, or the cell types are too divergent to successfully align without over-integration. It's important to note that this method is really new and there are still a lot of advancements to be made. So in a 2025 paper by Janida uses a new cross-species integration method called Saturn. Using Saturn, they were able to integrate cells that represent 686 million years of divergence. And after doing that, they were able to produce a cell phylogeny using the method that I'm describing here. So I'd like to incorporate Saturn in future pipelines, but it's not quite ready. So I'm going to present to you the results of my old pipeline. So using my original workflow, we found that I could not make a cell phylogeny from Planulozoa. I went a little more shallow and we looked at cnidarians, which is about 600 million years old. Again, cross-species integration failed, and I could not infer a cell phylogeny. So where we can infer a tree from 5 mammalian model species, we hit our upper taxonomic limit at 650 and 600 million years of divergence. But what about hydrozoans? You know, does my method work on anything that isn't a mammalian cell type? And it turns out hydrozoans occupy a phylogenetic sweet spot. They are closely related enough for cross-species integration to work, but they're divergent enough to still create a compelling and interesting cell phylogeny. So the first thing I found is that the pattern seen in part 1 is also true to these cell types. You can see that in this plot of the amount of variance per principal component. The data is low rank, and I had a big sigh of relief. And we also find that later PCs describe tip-specific variants, while earlier PCs may have the phylogenetic signal. Here, tip edge length increases, whereas interior edge length stays about the same. So this allowed me to produce a robust cell phylogeny of hydrozoan cell types. There are several stories here, but I'm just going to stick to one. So as you can see, well-supported clades of stem cells, germ cells, and other cell types came out, but there was something very surprising about this tree. So stem cells differentiate into somatic cell types. So if the stem cells in the somatic progeny are so closely related developmentally, they should be closely related evolutionarily too, right? But that's not what we see. Instead, this tree segregated by potency. Stem cells and germ cells formed a single highly supported clade, whereas all the differentiated somatic cell types formed a different clade. So germ cells aren't stem cells, but actually they're the most potent cell type in the body because they can give rise to every cell in an organism. So this is true even for certain somatic lineages that descend from specific stem cell types. So one example is the nematoblast. That's the stem cell that gives rise to nematocytes, which is the stinging cell type of cnidarians. So we can trace a direct developmental trajectory from these nematoblasts all the way to mature nematocytes. But on the tree, they don't cluster together. Nematoblasts cluster with the stem cells, germ cells, and nematocytes cluster with the somatic cells. So one interpretation is that although they arise from the same developmental lineage, the nematoblasts reflect an ancestral stem cell type, whereas the nematocytes reflect an ancestral somatic cell type. And as a result, they are fundamentally distinct evolutionary cell types. So this might actually have some very deep evolutionary implications. These findings may represent a fundamental evolutionary distinction that may have arisen during the origin of multicellularity. It's been hypothesized that before the origins of multicellularity, unicellular lineages were in constant competition. So this means that the unit of selection was at the unicellular level. For multicellularity to emerge, the unit of selection must shift up one biological level of organization. So it's cooperation between cells that shifted the unit of selection up. So this led to the emergence of multicellular individuality and selection at the level of the multicellular organism. But how did this happen? So this may have been achieved through the invention of the stem cell lineage. The stem cell lineage controls which cells directly progress to the next generation and which cells do not. So maybe controlling who wins may have been the mechanism that led to cooperation. So I propose that the stem versus somatic cell split that we see on the cell phylogeny could be a reflection of this ancient fundamental distinction. One more interesting thing I'll leave you with is that two types of stem cells did not cluster with the main stem cell germ cell clade. And these are the hydroectodermal and endodermal epithelial stem cells. Instead, they clustered with clades of ectodermal or endodermal cells. So these epithelial cells, they're not like the other stem cells, which are called interstitial stem cells. They look like epithelia and they can only give rise to more epithelia. So our tree represents the hypothesis that epithelial stem cells are convergent. Okay, so with my current method, we hit our upper taxonomic limit at 650 million years old. Our current cross-species integration approach is simply insufficient, but hydrozoans occupy a phylogenetic sweet spot. Hydrozoan cells also delivered a novel insight into the origin and evolution of stem cells. So this is significant because while mammals are only about 85 million years old, hydrozoans are about 450 million years old. Okay, so I looked really, really far apart at taxa separated by 650 million years. But what happens if I look really, really up close? Like, what is the narrowest possible taxonomic sampling I can do? Or how about a single individual? Like, part 1 looked at cells from a single organ across 5 species. Part 3 will look at cells from multiple organs from a single species, a single individual, in fact. So we have amazing databases of human cell type diversity. I decided to analyze cells from multiple organs of a single human donor. This donor was donor 14. He was a 59-year-old male who died of anoxia, possibly during surgery. So I want to take a minute to thank him. So donor 14 was chosen because he had the highest diversity of organs sequenced. He had about 12 organs sequenced, including things like the lung, the skin, the bladder. This data set gave me a rich opportunity to examine how the organ or tissue context may affect the evolution of human subtypes. So there's only one species here, right? A single individual. There is no species signal to obscure the subtype signal, so it should be easier to make a cell phylogeny, right? Well, those were famous last words. It was a bit more complicated than that. So here's my initial cell phylogeny of the data set. It's not a perfect tree. Subtype clades do kind of come out, but they're impure, they're unstable, and they're really star-like. I wanted to figure out exactly what was going on, so I plotted the cells by their principal component scores, and I examined how they looked in PC space. This is a lot of cells plotted by PC score and colored by cell type. And then I saw something kind of interesting. I found that clades in a cell phylogeny are embodied as cell strings. So there you can see strings of endothelia and erythrocyte and fibroblast cells. These strings formed when one PC tightly defines all the cells in the clade along a very narrow range on one axis, whereas the other PC broadly describes variants across the clade, and all of the cell types cluster around the origin. So I hypothesized that what we are looking at is a manifestation of the isolated cell type signal. So this is consistent with what we saw in part 1, where we found that cell type signals among gene ontology terms of highly loaded genes emerged. It seems that specific PCs might describe specific cell types. So this gives us actually a really neat opportunity. We can isolate the variants for cell type, pinpoint where exactly on the cell phylogeny it arose, and functionally investigate this phylogenetic hypothesis using the most highly loaded genes as candidate genes. So for instance, this is the cell type signal for neutrophils and macrophages that was isolated in PCs 8 and 9. PC 8 tightly defines these clades, whereas PC 9 differentiates the neutrophils from the macrophages. So I can hypothesize that this variance arose right here, right at the root of the macrophage and neutrophil clade. And among the most highly loaded genes were neutrophil-specific and macrophage-specific marker genes, things like MMDA, CXCR8. So one hypothesis we can have is that the expression of these specific genes drove the evolutionary origins of the neutrophil and macrophage clades, and we can now have a list of candidate genes to functionally investigate this phylogenetic hypothesis in downstream analyses. But I also found some organ signals, which are strings of multiple cell types that came from the same organ. So liver and tongue cells are an example. In this PC plot, the cells are colored by organ. So could the organ signal in early PCs contribute towards muddying the cell type signal, making it harder to make a cell phylogeny? Okay, so how do we study organ variants? Well, one thing you can do is take away the organ signal and see what happens. Just as you can integrate across species using cross-species integration, why not integrate across organs using cross-organ integration? So what they found is that removing the organ signal transforms the data to an extremely low rank. In other words, without organ variants, there is little shared variance left to infer the cell phylogeny. And you can see that here. I did a PC sweep of the data with and without the organ signal. And with the organ signal, phylogenetic signal in the dark gray steadily increases up to PC55, after which it levels off. But after removing the organ signal, the phylogenetic signal is overtaken by the cell-specific signal already at PC6. And honestly, you really can't infer robust phylogeny from 6 characters. So here's what it looks like if you plot cells by their PC score. Cell strings were present up to PC4, but after PC4, there was absolutely no structure to the variants, and all the cells cluster around the origin. So to return to our tally, where we can make beautiful cell phylogenies of cells that have undergone 85 million and 450 million years of divergence, we find that the lower taxonomic limit of my method is cells from a single individual. So we found the upper and lower taxonomic limit. 650 million years is too distant, and a single individual human is too narrow. So in parts 1 and 2, a narrow taxonomic sampling actually led to a better cell phylogeny. So these results were really surprising to me. But while taxonomic complexity was minimized, by looking across different organs, functional complexity was increased. So ultimately, I found that the organ signal seems to overwhelm the subtype signal in a single human individual. So maybe it's essential to analyze multiple organ cell types across multiple species. I hypothesize this can have 2 possibilities. This might either amplify the cell type signal independent of the organ signal, or if conservative organ level evolution is the rule, then maybe it'll just amplify the organ signal. But either way, functional divergence arising from unique organ contexts must be a powerful driver of cell type evolution. Okay, so what did we achieve? We present a method to infer the first cell phylogeny using an explicit model of evolution. We confirmed that it is indeed possible to infer robust trees from single cell count data, even though it's highly multidimensional and continuous. And we pushed the taxonomic limits of my method and found that there was a phylogenetic sweet spot. Species must be closely related enough for cross-species integration to work, but distantly related enough for the organ signal not to overwhelm the cell type signal. Okay, so my final section, we'll discuss how we use these tools to study a very explicit evolutionary phenomena, the origin and evolution of cancer. This will be my postdoctoral work in Selena Giuliano's lab, the molecular and cell biology department. Okay, so let me bring you back to the stem cell story. So we hypothesized that competitive evolutionary dynamics between cells occur during the origin of multicellularity. So multicellular individuality could only emerge when these competitive dynamics were regulated. So understanding these competitive dynamics might be key to understanding the origin of animals. But my biggest frustration as an evolutionary biologist is that there's simply no way you can step into a time machine and go back and witness these things in person. So does any type of cellular competition happen today? And the answer is yes. So cancer is a devastating but common type of cell evolution that happens rapidly, right in front of our eyes. Cancer occurs when one cell starts to cheat. It outcompetes the other and overproliferates, which leads to a tumor. Maybe if we study cancer in an early diverging animal, we can study competitive evolutionary dynamics that might have some parallels to what occurred at the origin of animals. So in the Giuliano lab, we study an extraordinary organism called Hydra. I've actually got a tube of them that you can pass around. So Hydra is a hydrozoan cnidarian. And if you remember from part 2, it has these special stem cells called interstitial stem cells. And it has amazing regenerative abilities. And that is due to the constant proliferation of these stem cells. So this is a video of Hydra. This is a video of Hydra that did form into a whole Hydra from a piece of its body wall. Okay, so Hydra has been studied for its amazing regenerative abilities for over 300 years, but it has another significant property, and that is a remarkable immunity to cancer. So Hydra vulgaris reproduces asexually in the lab, and it's been estimated that it could have a lifespan of over 3,000 years. In addition, its cells are constantly proliferating due to those special stem cells. So it should be absolutely riddled with cancer. But despite being grown for decades in the lab, we've never seen any cancer. In fact, no case of cancer has ever been observed in any early diverging non-bilaterian. No sponges, no placozoans, no ctenophores. And honestly, we really don't know why, but I hypothesize it could be because all these animals are highly regenerative. Okay, so imagine everyone's mind being blown when it was discovered that a closely related species, Hydra oligactus, spontaneously develops cancer all the time. So on the left is a healthy oligactus, and on the right is an individual with active tumors. The arrows point to the tumors. So oligactus appears to form its tumors in its germline stem cells. Each tumor shows many characteristics of vertebrate tumors. For instance, they can invade healthy host tissue, and they even express oncogenes that are similar to vertebrate tumor oncogenes. So this makes cnidarians the earliest diverging animal group in which cancer has been observed. So what I plan to do for my postdoc, among other things, is to perform whole body single-cell RNA-seq of cancer in oligactus. So I'll first compare this data set to hydrovulgaris to try to figure out what makes hydrovulgaris so immune to cancer. Maybe it has novel tumor suppressor approaches that would be of clinical value to humans. But in addition to that, I'll apply phylogenetic comparative methods using my cell phylogeny approach to directly study the evolution of cancer at the cellular level. So what can we do? Well, first we can investigate the ancestry of cancer cell types. Microscopy suggests that they arise from germline stem cells, but this isn't confirmed molecularly, and we honestly don't really know what kind of germline stem cells they arise from. So my approach would be to go to cell phylogeny, and the first clue will be the topology of the cell phylogeny. So are cancer cells nested within a specific cell type, or are there many independent origins of cancer cells? A second approach would be to be doing an ancestral character state reconstruction of the cancer transcriptome. So this here on the right is called a T state. It's a dimensionally reduced map of cells. Each dot is a cell, and cells that express similar genes cluster together. Each cluster is a cell type, and on this map, different cell types are colored in different colors. So we can map gene expression onto the tips of the cell phylogeny, and you can see that here, an example for one gene. And you can perform ancestral character state reconstruction to predict the expression level in the ancestral cancer cell. So if we do this for every gene, we can reconstruct the ancestral transcriptome of the ancestral cancer cell, and then plot it right back onto our map of cells to figure out which cluster it came from. Okay, a second question, can cancer cells undergo adaptive radiation? So we already know that these cancer cells invade healthy host tissue at a markedly increased rate. Are they encountering new niches? And when they invade tissue, do their evolutions have hallmarks of adaptive radiation? So again, we can glean part of this from the topology of the cell phylogeny. Does the cancer cell clade show a constant gradual branching pattern, or do they rapidly branch at the base of the clade, which would suggest irradiation? But we can also make this quantitative. We can use the cell tree to calculate tip speciation rates using the DR statistics. So now we can answer, does the cancer clade show a significant increase in speciation rate, or do they speciate at approximately the same rate as all the other cell types? Finally, let's study signatures of natural selection. So oncogenes cause cancer cells to grow more malignant and more aggressive. Thanks so much. Okay, so oncogenes cause cancer cells to become more malignant and more aggressive, and that's because oncogenes are genes that control proliferation, growth, and survival. In other words, they make cancer cells better competitors. So are oncogenes under positive natural selection? So cells don't exist in isolation in the body. They actually exist as populations. Coming back to the cell map, here each dot is a cell, and there's about 25,000 dots, and they cluster into relatively discrete populations. So the fact that cells exist as populations really hasn't been taken advantage of in single cell biology, but I know you guys in the Center for Population Biology do think about populations a lot. So maybe we can perform a classic test of neutrality on this population to figure out which genes are under selection. So for instance, maybe we can do something like a gene expression HKA test. So we can expect that the genes under selection would have low expression variance within cancer cells and high expression variance between cell types. And these would be our candidate oncogenes. So as you can tell, the phylogenetic tools I'm proposing here are pretty basic, but that's where we're at for cell phylogenetics. There's still a lot to expand upon, and I'm really hopeful that it's the start of something pretty interesting and pretty productive. So thank you for listening to me today. I'm in the molecular and cell biology department, but I'm an evolutionary biologist, and I would love to interact with you guys more. It'd be amazing to be an affiliate, or just a friendly face at a seminar. So thanks for having me, and I'd be happy to answer questions. Yeah.
Fascinating seminar, my gosh. I've never gotten so excited about principal component.
Oh, you should have seen me during my PhD.
So I'm thinking about your method, and it would take a lot more thought for me to completely wrap my head around it. I'll be honest, and so I'm hoping you can help me with something here. I think that your method is what I would call phonetic, and
Okay.
that your trees are distance trees.
Oh,
They measure similarity, overall similarity.
okay. Well, I personally would say no, but it's not your fault because I actually didn't present on the model of evolution that we used. So we used Fausenstein's RML PIC algorithm, and it's a model of Brownian evolution. And we used that to infer the tree, but I didn't present that. So yes, if I didn't do that, it would be phonetic.
But so explain to me how they're something more than a distance tree.
Well, with an evolutionary model, you can predict what happens, whereas a distance tree is just a description. Now, they might look similar because they both rely on similarity between phenotype, but they don't mean the same thing. Evolutionary trees model evolution. Distance trees describe evolution.
And so your tree is a Brownian, your model is Brownian motion.
Yeah.
Okay. Thanks.
Yeah.
To kind of follow up on that, how much do the divergent states play into that model? Do you use divergent states sort of a priori to inform the length of the tree?
I wish it was that advanced. They don't inform anything right now.
Gotcha.
Yeah.
When you're trying to sort of compare across species, like, it seems like paralogs would be a major problem for this, right?
It
So
could be.
I guess I'm wondering, like, how many genes do you actually get to look at?
So for simplicity, I did stick with single copy orthologs. I think that could be why it's really hard to look across really just the species, because eventually you don't share many single copy orthologs. But my character matrix had about 5,000 genes or so that were shared. Yep.
Yeah, really cool talk. I'm curious about at your larger taxonomic scales, do you ever see evidence of developmental system drift that might result in like thinking things are convergent or
Can you repeat that a little bit louder?
yeah, at your broader taxonomic scales, do you see evidence of developmental system drift that might kind of conflate the cell types versus the cell lineages?
Okay, that is a really great question. I have not looked at that. So I don't know.
All right, well, we're a little past time, so if you have any more questions, talk to Jasmine afterwards and let's give her a round of applause.